just at the nitrogen level. And also another objective we have or we are doing in this project is creating the tool. It's called intercrop tool, and later it will be converted into the decision support tool for the farmers, where farmers can put their the soil characteristic and the climate characteristic, the farm characteristic, and this support tool can give them different combinations of the intercrops. This can be used. So again, if I break down these meso objective, there is a more like macro objectives like selecting different diverse intercrop mixture, like evaluating the technical and practical barrier, creating the multi-actor approach, two-way communication between the stakeholders, especially the farmers and the researcher. And not just this, but also we have partners outside of Europe, so under in North Africa and also in Asia. So how there's a two-way communication communication with other part of the world. EU policies on the intercropping. Then what are the impact of intercropping on crop yield, quality, nutrient retention. Also, we are focusing on soil part, so how it can influence the soil functionality, soil microbiota, pest pathogen, greenhouse gas emission, soil organic, carbon matter, this, and then this decision support tool and the model. So these are the objectives of our project. I just want to give you a little bit about the information about the intercropping. I know many people know about the intercropping, but intercropping is simply growing two or more crops in a close proximity for a certain time of the period. It can be the whole cycle of the two plants, or at least the sum of the part of their growing cycle, they're grown together. So based on the plant arrangement, you can see here, if I can use the laser pointer. So there are how you are putting the plants inside. We have like strip intercropping. So you having some row of one crop type and then the another. So you have like 3 or 4 row of one crop and the other. And this is the strip cropping. Then relay intercropping is where you have, it can either be the single row or the multiple as a strip, but we are growing the plants in a different time. So they are growing together for not whole plant cycle, but part of their growth period. Then row, and then there is a kind of random, it's a mix. So you are just mixing the seeds and you are growing them. But there's also the intercropping have a type based on the seed density ratio. So but mainly we are using this replacement, where you just grow 50% of legume and 50% of cereal. Either the ratio can be changed 60-40, so you are just replacing. But the other kind is the additive, where in the same land you are growing more than 100%. So you are growing it can be either the 100% of legume and 100% of cereal also, or it can be 100% of cereal, 50% of legume. It depends how the farmer would like to go with this. So these are the different kind of intercropping. So what is the concept of leguminose? So basically the leguminose is based on the idea of ecological intensification. So in this concept, we are assuming that component of biodiversity can be used to either complement or even replace the artificial input. So if I say in other words that different species within a community, they are in some way, they are specialized and they can interact with their biotic and abiotic environment. And they're creating this division of labor, which result in the higher efficiency and the functioning of cropping system as a whole. So if we increase the biodiversity, we are hypothesizing that it increases the ecosystem functioning. So this is the basic concept. But later, the main hypothesis of our project is that you can see simply in this figure that when there is a monoculture and there is a intercropping, so we know that each plant have a different kind of root exudation pattern. Yeah, so through the root exudates, they are releasing different kind of chemical compounds. Microbes are attracted to these compounds. They use them for their own nutrients, and they mineralize these compounds and make nutrients available for the plants. So we hypothesize that when we are growing two plants together, there is a different kind of root exudation pattern between different plants, because we know that each plant have their own pattern for the root exudation. Even the exudation pattern change from morning to from day to night, and also from different phenological states. So here we are hypothesizing that there is a wider range of chemical substances are available, which can attract not the part of microbial community, but kind of the whole microbial community. So it can somehow improve the soil functional diversity. So there are more microbes attract, they can perform the more functions. So they will more nutrients will be available and available for the plants, and somehow it will have a positive impact on the plant production. So this was the main hypothesis we have in the Leguminose project. Okay, so I will not go through all the work packages and these things, but the project to tackle our main goals from the project from the different angle, from farm to value chain and to the societal level, and to coordinate our effort. The project is divided or structured into the different work packages. And they have they are answering the individual questions and the topics. But these the work packages are divided into 3 clusters, like research, creation of the tools, and the dialogues. So these are the 3 main components of our project: is the research, tools, and the dialogue. And as you've seen, that the project is a research and innovation. So we are focusing and balancing between both of the aspect of the project, the research and also the innovation. So, but both the aspect are very well synchronized to each other. In a research, what we are doing, we set up 7 research field trials in 7 European countries. And the researchers are the main actor, and they are filling the knowledge gap we have for this intercropping. On the other side, for the innovation, we have these living labs. We give the name living labs, but it's a kind of on-farm trials in 9 countries. So Italy, Spain, Poland, Germany, Czech Republic, Denmark, and 2 other non-EU countries, Egypt and also Pakistan. And each country we are establishing 20 on-farm trials with different combination of legume and cereals. And here we are setting these farms in a real condition. So the main actors are from the value chain and mainly the farmers. So we are directly working with the farmers, and we're doing this co-creation. And then the data are collected from the research trials and these on-farm trials. We gathering this data, and these data are used for the creation of the tools, knowledge gap at the research level, engagement, like stakeholder engagement, and the adoption of wider adoption of this technique. And these data will have or having the impact at a different level, the environmental, ecosystem, and socioeconomic level. So I'll show you some of the results we have until now from our research field trial, and also I will show you some insight from our on-farm trials. We already have 140 on-farm trials with farmers in a different countries. So as you see on the screen, we have 7 field trials. 4 trials are new trials. They're established in when the project started. And then we also have the 3 long-term trials in Poland, Czech Republic, and in Denmark. And they, like you can see in Denmark and in Poland, they are quite like more than 20 years. They have this field trial. So we can also understand that how the intercropping have an impact at the short term and also at the long term. So this is the benefit in our project. If you want to learn more about the trials and their like experimental design and everything, you can just simply go onto our Leguminose website and you can have more information. So I will not go like much about the other kind of data set. I will focus on the soil data set because this is the webinar is more about the soil fertility and soil health. So you can see that we have collected different kind of soil samples, like we have different VP. So we collected the soil at the topsoil and also soil up to the 90 centimeter. And we collected soil, also rhizosphere soil, bulk soil, and also the plant roots. And up to now, we almost collected from these field trials, more than like 3,500 samples has been collected for the analysis in 2 years of field trials. So what we doing with these samples? So first of all, we are analyzing the microbial community. So not just, so first of all, we doing the sequencing, like meta-DNA extraction and understanding the structure, composition, and the diversity of microbial community. Then how the network complex, like how co-occurrence pattern between different microbes. Also the functional diversity. So we are also looking into the different microbial genes which are involved in a different nutrient cycle and the carbon cycle. So nitrogen cycle, phosphorus cycle, and carbon cycle. So here I'm just showing the example of nitrogen cycle, and we are covering almost like all the genes which are involved in a fixation, nitrification, denitrification, all the things. Also, we are measuring like biochemical properties like microbial biomass, carbon, nitrogen, and in some sites also the phosphorus, different enzyme activities related to different biogeochemical cycles, and in some sites we also measuring the carbon use efficiency. And also we measuring different wide range of physiochemical properties also. What we will do with these, we are doing with all these huge data sets. So we are using these physical property, the measurement at the physical level, chemical and the biological level. They are used to calculate the soil multifunctionality, because the soil multifunctionality can give you, it is kind of an index to give it like for the soil fertility or the soil health. I will not go in a detail about how this functionality is calculated. We are just following the paper, Byron 2014, they proposed, and we are using the proposed formula to calculate the soil multifunctionality. So I'll show you some of the results from some of the field trials. So these are the different parameters we use to calculate the soil multifunctionality in different field trials. So you can see there are different enzyme activities, functional genes abundance, and also the like pH. And because we are still in the measurement, so we will update this data set and we will evaluate the multifunctionality again. But these are the very preliminary results. But still, from the preliminary results, what we found that you can see here that multifunctionality, either is the bulk soil or the rhizosphere soil, it somehow increased in the intercropping practice. This is the results from the Italian field trial, where we have a combination of soft wheat and the red clover, and this is an additive design. So we have in the same land, we have 100% of the ratio, the seed density ratio for the both plants. So it's in the same land, we have 2%, 200% of the plants inside. So you can imagine that there is a higher competition between the plants to uptake the nutrients, and still we have a higher soil multifunctionality in this field trial. We also observed the similar trend in our polish trial, where we have the similar kind of the trial, but this is the long-term trial, the spring wheat and the clover. And also here, but in this trial, we don't have clover as a monoculture. So we have a monoculture as only the wheat, and in the intercropping, we have a wheat and the clover. But still, you can see that the intercropping is higher. Sorry, the soil multifunctionality is higher into the intercropping system. Also, we found in a German field trial, there is no significant impact, but it's not decreasing the multifunctionality. The level is the same. So this is the short-term trial. So somehow we are assuming that if this kind of cropping system goes on, might be we will have a higher chances of increasing the soil multifunctionality in this system. And again, also we found the similar results also in our Danish field trial. Now I will show you some of the results from Italian field trial, because we're still analyzing the data about how this intercropping can have an impact on soil microbial community. So here, this is like principal coordinate analysis. We show you how the microbial community structure or is changing, or the diversity is changing under the different treatments. So if you can clearly see that in a bulk soil, rhizosphere, and roots, the intercropping, they have a significant impact, and they are changing the microbial community structure in all the kind of compartment, either the rhizosphere compartment or the rhizosphere or the root. But we found a very significant impact on the community into the root compartment. Also, we looked into the networks, how the co-occurrence network. These are the networks for the bacterial community. So you can see that it is obvious that from bulk to roots, the complexity of networks are decreasing, but also we found that the complexity of these networks are decreasing from cereal to intercropping. So cereal, the soil with the cereal crops have the higher, more complex kind of networks than the legumes and the intercropping. Normally, we finding that when there is a diversity, it increases the diversity and they increase the complexity of the microbial co-occurrence network. But what we are hypothesizing, because it is still we are analyzing the data, but what we from our understanding, we can conclude that in the intercropping might be there is enough amount of the nutrients available, so there is a less interaction, or there is a less competition between the microbes. That's why it's reducing the interaction between this microbial community. But still, we don't know. This is just the data from the one side. We have to evaluate other data set, but this is the preliminary evaluation we have. What I would like to show you later further, just to confirm, we also have kind of some of the unique kind of nodes are like taxa, which like microbial genus or the species. So we found here that there is only very few number of the taxa are shared between different networks between even also in the different compartment, the bulk, rhizosphere, and roots, and the edges are like how they are interconnected. So there is a very few amount of similarity between different networks between the cereal, legume, and intercropping. So the microbial community is changing, and the intercropping have an impact on the composition and the structure of microbial community. Then we also we compared that how stable these networks are, and how you can do this. So you can just randomly remove some nodes like a taxa into the network, and you can see that how these networks are connected or stable. And what we found that, okay, we have obviously when you remove some of the taxa or the nodes from the network, they will become distable. But still, if you see here, between compared to the monocultures, either is a cereal or the legume, in an intercrop, networks are little bit stable compared to the monoculture networks is. So what I'm trying to say is that if there is a stress condition, the networks or the interaction or the microbial community present into the intercropping can be become tolerant or become resistant to this stress. So they can be more stable under the stress condition. This is what we are assuming from our results. And also we calculated the yield from different field trials. And what we found that there is no negative impact, or there is no low yield in intercropping. It is more or less the similar yield compared to the monocultures, or in some cases, the yield is increasing. So the intercropping has a positive impact on the plant yield. But this plot is showing that the yield is significantly correlated with latitude. Yeah. So what we found that the yield, when the climate is warmer, the intercropping has a higher impact, the positive impact on the plant production. When there is the little bit colder kind of environment, it doesn't have a negative impact, but even it's a kind of neutral impact. It's not having a negative impact. So even if you're doing the intercropping, it will not have a strong positive impact on a yield, but somehow it can impact your soil fertility. Okay, now I would like to show you some of the results we have from our on-farm trials, the living labs. So as I mentioned in the earlier slides, that we established almost like 20 trials in each participating country. And until now, we have 140 coordinates. Like we have established the trials in almost 140 trials in the country. So you can see these blue dots are showing where we have our living labs. So this is in the Europe, it's Egypt, and also in Pakistan. So then we collected the data from these trials and we checked different aspects. So obviously, not all the trials were successful. Some of the trials were failing, and there are many reasons. But what we found that compared to the cereal, we have more failing into the legume crops. You see here, it's almost double amount of the legumes which are failing into either it can be the monoculture or the intercropping. And then also we checked what are the reasons for these failing. So there are diverse reasons, like in some trials, there's wild animals, they attack. In some, there's weather or the disease, termination of cooperation, like farmer doesn't want to do it, and also technical failure. So there is many parameters. We found that these are the reason for failing of the living labs. And we found that in all the countries, in 140 trials, we found that what are the selected crops by the farmers. So we found different 15 varieties of cereals has been selected, and 17 of the legume, and different combination of these variants has been used. And in the end, we have almost different 45 kind of intercrops has been used in our these 140 trials. And then we also looked at which kind of the farm or the soil management. So higher managements are arable production, but also we found some of the trials into the dairy production, livestock farming, or the mixed kind of farming. But most of the trial, like almost 50% of trial were conventional. But we are happy to see that almost 21% of the trials are organic, and up to 8% are integrative, like part of conventional and the organic mixed together. Some of them are with tillage, no tillage. So we have a diverse kind of soil management practice also. So also we found different kind of intercropping has been used, like alternate or the mix or the strips. And when how these intercrop products has been used, like it has been used by different industry, like the fooder or human nutrition. For human nutrition for fooder, then especially for the human nutrition industry, specific to the industry. So we have a different use of these intercrop after their harvest. But also what we found, the similar trend we found also into the yield. We calculated the land equivalent ratio, and what we found that the climate has a strong impact on the yield in intercropping. And what we found that when there is a warmer climate, it has a positive impact. And if it is the climate is becoming colder, it has a neutral impact. Not the negative, but you can see from this plot, but still the climate is one of the main factor in this management practice. We also have another aspect like understanding the different hurdles or for this adopting or the adoption of intercropping. So kind of socioeconomic analysis. So we ask people in the 9 countries what they think about the intercropping by SWOT analysis. And also we conducted a survey, and we collected almost more than 2,000 survey to understanding what are the barriers and the opportunity of the intercropping. I will not go much deeper into this. We have a published paper also on this one, Stone et al. It's available on the website of Leguminose project. You can just read it about this paper in the detail, and we will have more papers, more publication is coming on this data set. But still, we find there are some hurdles in adoption of intercropping, not at the management level, but at the social and economic level. So what I can say to you as the take-home message, that intercropping have a significant impact on soil fertility and crop production from our preliminary data. Is not like the final answer. And it can be used or it can be employed as a climate smart farming practice, because we know that due to the climate change, the temperature will increase, either also in the northern part of Europe. So I think it can be used as because it's giving a positive correlation with climate. So it can be used in the future as a climate smart farming. However, we still found many challenges, social and economic challenges. For example, like during the most complex challenge is the harvesting. Yeah, how to harvest 2 plants together, how to separating them, and how selling these products. But we made some progress in this. So I think in future we will also have a solution at the social and economic level for adopting these intercropping practice. So that's it. I would like to thank is not only my data, is the effort of the whole consortium. So I would like to thank first of all the official coordinator, Giacomo Pietramellara, my mentor, my team in Florence, and also all the people. There are few of them here in the picture, but it's a huge consortium. So I would like to thank all of them. Thank you. And thanks all of you for listening.
Perfect. Thank you. Thank you very much, Shamina. This is like it's a huge amount of information. If you want to know more about the project, I just put the website in there. They have a very nice resource fact sheets with like small facts about the projects and the field trials that they have been conducting. It's very interesting to read that bit. We have questions coming. So the first one is regarding the bacterial co-occurrence in the different cultures. Could the symbiosis in intercropping hide the diversity, for example, not seen as different or unique microbial cultures any longer? Uh, and the second question is, you say bacterial co-occurrence, but I suppose you also maybe sequence for fungi.
Yeah, we sequence the bacterial community and also the fungal community, but we're still analyzing the data. Yeah, so I didn't have a time to create the network analysis for the fungal community. So we sure with the data we have the data and we will analyze these networks co-occurrence pattern into the fungal community also. And uh, I think from the network here you don't see the much information about the which texts are present there, but we have this information and we will analyze this network further, and we will understand we will also find out with the statistic the kind of keystone taxa into these networks in a different practice, like in monocultures and also into the intercropping. But I don't think so they will hide this symbiosis one, but because that's why we did sequencing also in the root samples, because so I think we will see it, but I don't think so it will hide. It can increase, but also from the composition, we found that intercropping is increasing the rhizobia abundance also in the roots of cereal. But this is from the very in the first year results, but we have to analyze all the data to confirm these results.
Okay,
I hope
excellent.
answer.
Yeah. There is another question here. It says, do you know if intercropping is affected by global warming? Which I think you touched a little bit in there. So the person is asking specifically in maturation of intercropping legumes, maybe earlier in the season due to warming, such that harvesting needs to be earlier and may not align with the harvest date of the main crops anymore. Are you aware that this is a real world problem? And if yes, what do you think can be do to solve this?
Okay, so it's really okay. I'm not the agronomist, yeah, so I will try to answer the question. Okay, so sometime it happened that some of the plants, like one of the plant we growing, it reached to the maturity earlier. So you have to harvest earlier, but it's really depend on the design you used and the machinery the farmers have, In some of the, like in a strip intercropping, you can harvest that plant and you can still let the other plant grow and you can harvest later. So this problem, I think, is coming into the row intercropping or maybe mix intercropping, yeah. And which kind of intercrop combination you are using and what will be the use later. For example, if you are using you are growing the clover as a companion plant, and just you don't interested in the yield or harvest of the legume like forage, then you can harvest them at the same time. But I think it is the problem, and also there will be the competition in the water uptake. But there are still many problems, but we are trying to tackle. Let's see
Right.
where
Yeah.
it's going. Yeah.
Yeah. We have a group of questions here.
Yeah.
It says, are you looking to use these intercrops once they are harvested? And when they are harvested, they can be harvested fully developed or still in a green stage. And then a second part is that because I had a project on this, but I did
Mhm.
not focus only on the plant side. I went also to use the intercrops to create value chain.
In our case, most of the harvest was done at the maturity stage. This is it. But some of them, I say that there was a fail. Some of the trials were failed because of the weather, especially in what I call the spring crops we used in Tuscany. One of the example we used it is the maize and the soya bean, but we had a very heavy rain during the spring and also into the autumn. So the trial was failed and we were not able to harvest the intercrops. So yeah, but it's really depend on the, is not, we cannot generalize this thing, but in most of our trials, we harvested at maturity stage.
And then we have from the same person, do you use always the same type of plant arrangement throughout the trials, or are they different according to the place where the trial takes place?
I already showed one slide that we used a wider range of intercropping trials with the like strips, row, and the mix. So we have a different combination. It really depend. We left this decision on the farmer how they would like to do because we would wanted to test this in the real conditions, you know.
Mm-hmm.
So farmer decided how they wanted to do in the farm. So we have like different combinations in a different trials. So it's not just specific to one country. In a different countries, different farmer use different kind of arrangement.
Mm-hmm. And we have many more questions coming, but I'm just gonna close with one last one, and it's about you coordinating these big projects. So, what advice would you give to young researchers who are planning to undertake a coordination of a Horizon Europe project in your experience?
Okay, it's a stress. It's a nightmare to coordinate the price. Like you are hurdling the chickens, you know, you have to run behind them, always answer questions. So if I'm not too early career researcher. I look young, but I'm not too young. So I'm in the middle of my career. So if you're a very young researcher, I give you advice that at least start understanding clearing your mind with what kind of research questions you have to develop in your career. And when you come to in the middle of your career, and it depends on the opportunity. If you have the opportunity and if you are confident, you can do in early stage also. So
Yeah, and I imagine your mentor also like played an important part, like having the
yeah,
support of a mentor there for you
that's
and
true.
a good team that is
Yeah.
willing to put the, the work, like hard work.
Yeah, I already mentioned that official coordinator is Professor Pietra Melara, but he put me in the front. That I wrote the project. It was idea, so he said, "Okay, you lead it." So it's also depend on your mentor and support from your team. Yeah,
That's great. That's great. So everyone else has many questions, so you can go into the Leguminous website and your email is there. So if they want to
yeah,
reach out to you and ask the questions, that's perfect. So
yeah.
I just wanted to try one last thing, because I want to ask people. I did a poll. I want to ask people what do they think they would like to see in the next series. I'm just gonna try this the first time. I don't know if it's gonna work. So I'm gonna launch this poll. It's about the next series that you think uh, it could go into the webinar series. I don't know if the poll is showing. Oh yeah, I can see it's populating. Interesting. It's the first time I do this. So basically, the poll shows in your screen,
Yeah.
and you can click on it. Nice. Okay.
I
Okay, we
see
have.
many questions, but you can just write me email. I'm also on LinkedIn and Twitter, so you can also message me directly there, and I'm happy to answer all your questions for the project, for the collaboration. We are open to the any kind of collaboration, so you can just write me.
Yeah, that's fantastic. So thank you very much, Shamina, for this talk. And I was very happy to have you with us to be able to get you because I know you're very busy with these projects and probably other projects. So thank you everyone for your questions and for your responses to the poll. And we're gonna have a break for the summer, and we'll be back in with a with the other talk is going to be sustainable renewable energy value chains for answering farmer needs, which is the project Value for Farm. That's going to be on September 3rd. So I wish I'll see you everyone there and have a wonderful summertime. Thank you everyone, and thank you, Shamina.
Thank you. Thank
Goodbye.
you. Bye-bye.
Bye.